Okay, the next speaker is Janneke Schwaner. She's a postdoc at the University of California, Irvine, working with Professor Monica Daley. Janneke's research focuses on the biomechanics of organismal behaviors, and today she's going to share with us some recent advances she made. We are very much looking forward to your talk, Janneke.
Well, thank you for that intro, and thanks all for having me today. So generally speaking, in my research, I aim to further understand the neuromechanics of extreme organismal behavior, like jumping or locomotor agility. And so far, I've been using kangaroo rats and guinea fowl as model species of bipedal locomotion. And today's talk, I want to cover briefly some projects highlighting some actuation, design principles, and control in animals during extreme performances. And I'm glad Talia already told you all about tendon ability to store and recover energy, as I'll be talking about some other roles that tendon can play in locomotion. And I would like to start off with talking about jumping kangaroo rats, and despite they may look a lot like jerboas, it's a very different animal. So kangaroo rats are bipedal hopping desert rodents native to Northern America, and they use vertical leaps to out-jump strikes by snakes, propelling themselves up to over 10 times their own hip height. And for comparison, if scaling worked ideal, that means you and I could jump over a 2 to 3 story building. And while these animals are airborne, these animals change orientation to take off in a newly acquired direction immediately after landing. So when seeing these videos, I wondered, how do kangaroo rats evade predation by snakes? And to answer these overall question, I had 2 sub-questions, and these will lead you through the presentation of today. So my first question is, what are the mechanics of kangaroo rat escape leaps? How do these animals propel themselves up so high? And the second one was, how do kangaroo rats use their tail to escape during these escape leaps? And to start off, we have to look a little bit more into detail of how these animals are built. So as in most cursorial animals like horses, goats, rabbits, but also guinea fowl that will pop up later in this talk, the majority of the kangaroo rat, here on the right, its musculature is located proximally or higher on the leg. Distal limbs of these animals are mainly made up from bones and tendinous structures, as Talia already told you all about tendon. And remember that muscles are the only actuators in our movement. Consequently, many more of the muscles in the hind legs are acting on the hip and the knee joint compared to the ankle joint. Based on this, one might expect that most work is done at the knee or the hip joint. However, eventually the work has to be done against the ground, otherwise animals cannot propel itself up. So we asked our kangaroo rats very kindly to jump over barriers, and for each jump we calculated work at each hind leg joint, as we can see here. So on the y-axis we have our work in joules per kilogram, and each vertical combo of couple of 3 dots, blue, an orange, and a pink, it represents 1 jump. And we found that most work is measured at the ankle joint that you can see here in blue. And this is the case even when jump height increases. These results suggest that the relative contribution from the knee joint even decreases when the jump height increases. Since the animal has more proximal leg muscle, it is not what you would expect based on anatomy. But there are ways to get energy from more proximal to distal joints. And one way is transfer. So the main ankle extensor, the lateral gastrocnemius, is biarticular, which means that the muscle tendon unit crosses more than 1 joint, in this case the ankle and the knee joint. It also means that energy can be transferred from proximal muscles due to the extension of the knee by this articular linkage. But how? So the stick figure on the right gives a very straightforward representation of this. So if the red, the muscle and the tendon unit, is stiff, which is the case as well for jerboas that Talia talked about, as well as for K-rats, moving this upper part of the leg, the femur, the energy is being transferred over this linkage, and the ankle joint will extend and the foot will be forced down. With the use of muscle movement arms, joint angles and the tibia length, we calculated the MTU length, and ultimately we obtained the work transferred over the biarticular linkage. And when we now go back to that joint work figure, we now see still the blue is still the same angle as we saw 2 slides before, but now we don't show the hip or the knee joints, we just show the work done by the muscle tendon unit, so that is the LG muscle and the tendon, and the proximal transfer. And we can see that in kangaroo rats, up to 40% of the work that we measure at the ankle is actually coming from our proximal muscles. But this made us curious what muscles, the only motor in our movements, are actually doing during these jumps. And because of muscles' unique anatomy, they have an operating range at which they can produce the most force, as you can see here in the force length graph, where sarcomere lengths are plotted in relation to the force producing capacity of the muscle. However, there is another relationship that is key for muscle performance, and that is the force velocity curve, here in red. So the gray shading, a little bit arbitrarily placed, but the idea is that it's around the peak of the blue curve, which is the power curve. But you can see that this gray shading indicates an optimal range where the muscle can produce most power. And as power is the product of force times velocity, these graphs are highly interlinked. So, and this brings me to another advantage of having less muscle and more tendon in the distal limb. And to illustrate this, we recorded in vivo shortening of the lateral gastrocnemius and calculated the shortening of the whole muscle tendon unit. So the figure on the right shows the lateral gastrocnemius muscle in dash and the whole muscle tendon unit in solid over an entire jump. And we can see that the muscle tendon unit shortens more. And elastic structures in general cannot shorten by themselves. However, when you have an elastic band previously stretched, it can shorten. And this is important to notice when looking at muscle shortening versus muscle tendon unit shortening. And to show why this difference in shortening is advantageous for muscle, I'll pull up the next figure. And we see here this lateral gastrocnemius, our calf muscle, the strain rate, so the shortening velocity, the ankle angle in the small dashes, and the angular velocity of the ankle joint in the big dashes. The gray shaded area that runs horizontal over the graph indicates this more optimal range of shortening velocity, or that identified sweet spot where muscle wants to operate to produce most force. And we can see that the black solid line passes through this spot for a little while, meaning that the muscle at that time operates as a more optimal condition. And when we compare the time this muscle operates in the sweet spot over all trials, we can identify that time range in which the muscle operates in general over all jumps. And this is visualized by that bar graph that you see below. This means that the relatively long tendon that is attached to the LG, to us known as the Achilles tendon, facilitates optimal conditions for shortening in this muscle. But as the relationship of muscle lengthening and force indicated, it matters to the muscle when to have these optimal conditions for it to be effective for power output. So when we plotted this third graph where we can see the ground reaction forces, we can see that the tendon facilitates more optimal conditions at times of higher force. So how do kangaroo rats evade predation by snakes? And specifically, what are the mechanics of kangaroo rat escape leaps? I hope you got from this that the musculotendon anatomy enables a transfer of energy from proximal muscles, and it allows muscles to operate at more optimal conditions. But when studying these jumps, I got fascinated by the entire escape leap, and I wondered how important these tails are actually that we see sweeping around for stabilizing or maybe even reorienting in the air. So for this project, we hypothesized that if the K-rats use their tail to reorient in the air, we expect to see in the data that the angular momenta of the tail and the body should mirror each other. And to test this hypothesis, we had to haul a bunch of equipment through the desert, which looks like this. Probably not the best day to put my pink t-shirt on. And despite it took a lot of time, we did record over 250 events, but I'm only going to share one. So here you see a K-rat from 4 different views escape our fake rattlesnake. I'm not a fan of snakes, so we used a fake one. And from these videos, we obtained three-dimensional kinematics as we digitized 10 points on the tail and a handful on the body to determine body and tail rotation, angular velocity, and eventually angular momentum. And what we found was that the reorientation of the body is proportional to the reorientation of the tail, with a regression slope of 1, which suggests that the kangaroo rat used their tail to reorient while airborne, supporting our hypotheses. And currently, in an active collaboration with Stacey Shield and Amir Patel from the African Robotic Unit at the University of Cape Town, we try to understand, through inverse kinematic simulations, based on their field data, how these animals use their tails for reorientation. And today I can provide a short teaser of what we found. And for this, I will solely focus on the yaw angle. So kangaroo rats try to land facing the direction they are jumping. And we can see in this graphic representation that most animals land within 45 degrees of the velocity direction. And if we then look at what the effect of the tail is for all these jumps, we can see that the tail has a corrective effect on the yaw angle during these escape leaps, illustrated by most of the dots, so each representing a single jump, to be located under this diagonal line. There is definitely more exciting stuff on how these animals use their tail, but you'll have to be a bit patient until we've fully written up this exciting story. But in conclusion, our data shows that kangaroos actively use their tails to assist in reorientation. We are still continuing our work to describe tail effects on pitch and roll axes as well. So now I would like to shift gear a little bit to work that I've been doing here at UCI using guinea fowl. So my research here focuses a little bit more on the central question of how do we use our neuromusculoskeletal system to navigate terrain. And to discuss some experimental methods to answer this big question, I want to talk to you all about two different projects. One focusing on obstacle negotiation, and one where I asked the question, how do birds navigate speed perturbations? And I'm interested in rough terrain navigation because it's complex, and yet under healthy conditions, most of us are really good at it. So generally speaking, navigating through the world around us requires a dynamic interactions between the environment, the sensory system, and the musculoskeletal system. And in the lab where I'm now, we use guinea fowl as a model system to investigate how we experience and respond to changing environmental demands while navigating the world around us. And motor commands arise from a synthesis of feed-forward planning. For example, I see a stair, so I'm going up the stairs, and reflex-mediated feedback corrective responses. So, whoa, I just missed a step, or maybe you thought you missed a step, but you didn't. Or you're about to miss a step. So for this particular project, we're interested in control. And to be more exact, we're mostly interested in peripheral reflex mechanisms. And to explain how we study these birds' muscle mechanics and sensory mechanisms, we have to look at how commands arise. So here we have our whole bird with a brain, spinal column, and a peripheral nerve leading to a limb muscle. And in this case, yes, it must be my favorite muscle. It is again the lateral gastrocnemius, or LG, which is the main ankle extensor. And if we now look at how commands arise through an animal system, we can identify that there is sensory motor networks and mechanical responses. And these types of responses appear on a different temporal scale, in which you can see that visual cues gets processed in 1 to 3 steps, dependent on locomotor speed. But most mechanical responses are typically really fast. So if we look at running, so here we have a guinea fowl running on an obstacle treadmill, with simultaneously running a work loop in purple, which is a graph where we have length, so muscle fascicle length on the x-axis and force on the y-axis, and then we have separately muscle force and EMG in the lower graphs. And from these studies on these running birds, we know that when running, you're more reliant on intrinsically stable mechanics and feed-forward muscle activation, especially during obstacle terrain navigation. Whereas during walking, birds are expected to be more reliant on anticipation and reflex modulated changes in muscle activation from stride to stride, based on these differences in time availability. And one way of studying these reflexes more rigorously is by inducing a proprioceptive deficit, which essentially gets rid of the sensory feedback from within the animal, so from the leg to the head, which have been successfully induced through self-reinnervation procedures. So for the current study, we hypothesized that if walking is inherently more reliant on reflexes than running, we think that reinnervation should result in a delayed recovery response due to the sensory deficit. And to test this hypothesis, we looked at muscle activity, work, and force in walking birds while navigating obstacles on the same obstacle treadmill you saw a couple slides back. But to talk you through this, we also categorized strides, and we identified strides based on the instrumented leg, and strides were cut from based on the walking cycle from mid-swing to mid-swing. And we categorized 4 strides. We categorized a pre-obstacle stride, a stride where the instrumented leg contacts the obstacle, and a first and a second recovery stride. And I'm going to talk you through 3 graphs to show you what we found. And they're all going to look the same, but I'm going to walk you through the first one, then the other ones make more sense. So here we see the EMG activity, so it's the muscle activation. And the gray bar is the mean difference, plus minus 95 confidence interval, between the surgical treatment conditions. And for every stride category, so here we have the S-1, so the stride before the obstacle, we have 2 bars, which in blue is the intact and orange is the re-innervated birds. And then we have the obstacle stride, and all obstacle strides will have a nice blue area behind them, shaded area. And I immediately want to point out here that the re-innervated birds show a smaller increase in EMG activity in this obstacle stride. And then we have 2 recovery strides, and we see here the exact same thing, is that there is a larger increase, more muscle activation during these recovery strides. And then we look at muscle force, where we also see a larger force increase in the obstacle stride as well as the recovery strides. And at last we have work, and this is surprising to us, we actually see that there's a much smaller increase in muscle work during the obstacle strides. And therefore we wanted to look at kinematics to see what's going on. And to investigate what the dynamics of the center of mass is doing, we plotted group average trajectories from the center of mass height. So here for intact birds during level walking, the down pointing triangles indicate the time of foot on, and the upward pointing triangles indicate the time of foot off. So interestingly, if we now look at the re-innervated birds, we see that these birds have a lower center of mass height, even during level walking. And if we now look at the obstacle strides, so here in blue for intact and orange for the re-innervated birds, we see that the intact birds lower their center of mass height during obstacle encounters, but relatively speaking, much more so than the re-innervated birds. And when looking at the second recovery stride, for reference, we see the level data in gray, but we can see that the intact birds recover fully before the second recovery stride, whereas we still see a little bit of a difference in these re-innervated birds. So in summary, we found that the re-innervated birds showed a delayed obstacle recovery in walking, consistent with our hypotheses, that the gastrocnemius muscle increased force but not work in obstacle encounters, shifting to more isometric contractions, and birds adjust to a more crouched posture following a re-innervated procedure, all suggesting that proprioception and sensory feedback is key to recovery. It also suggests a shift in strategy to either minimize the work of obstacle negotiation or that they rely on other muscles, such as more proximal hip and knee extensors that contribute to the work by transfer over bi or multi-articular joints. Additionally, our data shows that the birds increase stability by adjusting their posture, which we didn't see happen in running birds. Despite these birds, the walking and running cohort were the exact same birds, highlighting that stability compensation is speed dependent. And this made us wonder how we deal with perturbations that we cannot see. As for those, you solely rely on muscle mechanics and sensory feedback. And that brings me to the last little part of this talk, where I want to talk about blindfold bird. Oh, okay, more accurately, non-visible perturbation bird. As I've shown before, there are multiple contributors to control, and as we use treadmill belt speed as a perturbation for the project I'm going to describe now it eliminates the bird anticipation, which allows us to focus on other components of control. And for today's talk, I would just like to talk briefly about some data on the evidence of mechanical control, which is hypothesized to be particularly important for the distal joints with highly elastic musculotendon systems and fewer actuators or muscles. To help frame the interpretation of joint function in terms of intrinsic mechanical control mechanisms, I would like to talk first about general mechanical characteristics of locomotion. And generally speaking, and this might look familiar if you've seen Talia's talk, bipedal walking can be modeled as a spring-loaded inverted pendulum in which the leg is represented as a stiff compressive spring during stance. So high leg stiffness results in a trajectory in which the center of mass of the body is highest at mid-stance, and there is a passive exchange between kinetic and gravitational potential energy. Leg angular cycling and leg length actuation present the two basic modes of leg movements that influence the motion of bipedal gaits. So BirdBot, a recently developed bipedal robot, mimics this bird-like multi-articular muscle tendon linkages and includes an elastic latch mechanism in the distal joints that locks the leg spring into a high stiffness state during stance. Stored elastic energy is then rapidly released at the end of stance. And by using this elastically actuated mechanism at different speeds, BirdBot can walk and run economically. So we hypothesized, after we saw this video, that guinea fowl use a similar elastically actuated mechanism in walking. And if so, the distal joint kinematics and the timing of stance swing transition should be dictated by the rate of leg angular cycling, like we see in BirdBot. So to test this hypothesis, we varied the treadmill belt speed. I see my video is not running that smooth. These birds walk much smoother than you see here now. But we varied the treadmill belt speed to elicit perturbations in foot speed and leg angular trajectory without inducing changes in joint posture or gravitational loading. And we found that the belt speed perturbations, independent of perturbation category, influenced the rate of leg angular cycling via the hip angle and the timing of swing and stance transitions. However, the trajectories of joint angle, leg length, leg angle, and the center of mass oscillations remain similar. This suggests a very stereotypical kinematic pattern that can just be played slow and fast. So detailed analysis of in vivo muscle data we collected over the past year will allow us to answer our question of what acts as a lock in the spring of the guinea fowl leg, how birds used our neuromusculoskeletal system to navigate speed perturbations, and if there are differences regarding locomotor speed or surgical condition. The analysis of the in vivo and in situ data collected for this project is underway as we speak. So for today's talk, I revealed some aspects of how animals jump, reorient, and navigate through rough terrain with the use of two bipedal animal models. I also highlighted some form-function relationship of hind limbs and tails, as well as some aspects that are important for control. Because understanding muscles as complex multitasking organs in movement is important for interpreting the remarkable diversity of musculoskeletal form and function among animals. And with that, I would like to thank my lab and my lab mates and my funding, and you all for listening today.
Thank you so much, Janneke. It was super interesting. So if you have any question, please either write in chat or press the button raise hand. So Steve, is there any question there?
Yeah. You can just grab that one, I guess. I think you have to turn it on.
Hey, it's Scorpion again. So I'm also doing the biomechanics of human body. So I have a question regarding the biarticular muscle, the gastrocnemius. So a general question would be that, did you find in the guinea fowl or in the other animals, like the functionality of the biarticular muscle, like the gastrocnemius, and functionality of the monocular muscle, like vastus or soleus? Are they actually, like functionality depends on the task? For instance, if you have a periodic motion, like a continuous hopping, and if you have a sudden jump, for instance, do they have the similar, like for instance, the work contribution to the whole like center mass height in the jumping case or not?
No, I think that we know that the actual role or function of a biarticular or multiarticular muscle mainly depends on function. The thing that I also think is really important to acknowledge that humans also have like a very big uniarticular calf muscle or soleus, whereas I think that that gives kangaroo rats kind of a pre as a model species because they have barely any soleus muscle, and that's why it was so more straightforward to calculate how much work was actually transferred over this biarticular linkage, because we knew that there was barely any work coming from the soleus muscle that we were measuring at the ankle joint. But I do fully agree with you that it totally depends on function and task.
Okay, thanks. I did not get that part. Like you said that the soleus in the human muscle, in the human body is quite large, but there's almost no soleus in the guinea fowl, for instance.
No, in the kangaroo rat. So kangaroo rat barely have a soleus muscle.
Okay, I see. But do they still have like the tendon, like Achilles tendon, similar to the human body?
Yeah.
If so, then the Achilles tendon attached to the the monocular muscle should also kind of like functions as, for instance, a spring storage system in the periodical motion or not?
Yes, but to a lesser extent than in humans, because kangaroo rat tendons, Achilles tendons, are more stiff. Less stiff than we thought in 1980 when the first papers came out on their stiffness, but they're more stiff than our Achilles tendon.
Okay, thanks. And another question regarding the perturbation experiment you did with the guinea fowl. So it basically showed that, if I get it correctly, so basically you cut the neurofeedback from the gastrocnemius to the spinal cord, and the result shows that the work from the gastrocnemius is kind of like smaller compared to the intact birds, right?
Different. The muscle takes on a different role, and I think that is a very important one. So what the muscle does, it operates more isometrically, which makes the muscle act more as a strut, and that allows more work transfer from the more proximal hip and knee muscles.
Okay, so that's kind of surprising for me, if I recall correctly. So there's, like, for instance, in the study from the step down perturbation for the guinea fowl, right? So, by the way, do you think these two different perturbations, one step up or step down, they would have similar effects on the gastrocnemius or not?
I think you refer to one of Monica Daley's papers from 2011, where the bird does a invisible downstep.
Right.
A, that's a very different task, and B, those were intact animals. No, they didn't have a reinnervation procedure.
Thanks.
Okay, great.
There are a couple of raised hands in the
Zoom
audience.
Yeah, Talia, you want to go ahead?
I think Joao was first.
Okay.
Yeah, so mine is more of a curiosity. So I think that you presented like a lot of really cool abilities that these animals have with muscles. And I think that from a control perspective, muscles suck, right? Because they're incredibly inefficient. They have crazy nonlinear properties. They're super slow, but somehow animals are really good at using them. So do you think that we just don't know how to do proper controls, or do you think they're actually good, or do animals figure out a way around it? What is the trick?
Oh, that's a big question. Well, I do think that muscle from an engineering perspective might suck. It's probably true. But I think that one of the big aspects that muscles do and that are highly underappreciated is that muscle is not only a motor, muscle is also a sensory organ. And I think if you see those two together, that muscle becomes a much more interesting organ, and especially together with the tendon. And maybe like I haven't even like brought that message home hard enough during my talk, but the tendon is, even in a biologist's eye, at least in a comparative biomechanist view, Talia, tell me if I'm wrong, but tendons are relatively stupid springs, but a muscle can make so much more out of it, because a muscle can make the whole muscle tendon unit a strut, but it can make it also a spring, and it can also make it a brake. And I think that you can so quickly switch mechanical function within a single stride, even, that makes animals so absolutely awesome at maneuvering, because they technically don't care, because their body will tell them upon touching the ground what to do, and it's all unconscious. At least for a large part, it's unconscious.
Right, thanks.
Tendons are also sensors too.
Yeah, that's what I'm saying. Like they're,
Yeah.
sorry. Like, yeah, if we start talking about muscle, I can go forever.
So Talia, you want to go ahead?
Sure. Yeah, this was a great talk, Janneke. And I was wondering about the postural control for stability. And I'm wondering, how broadly do you think that kind of strategy can work? Is it just viable in guinea fowl, or do you think that if any kind of bipedal legged system wants to have that increase in stability, should they do this kind of postural readjustment? And then if it's so stable, why would you ever want to have a different posture?
Okay, I felt a couple of questions there. So I think that the lowering your center of mass and being more stable, I think that is something we do see in humans as well. So if some humans have like nerve deficits, they make their step width wider, or they like set their feet further apart. I mean, if you stand in front of me and I say, well, go stay stable now, what you do is you actually set your feet out a little bit more wide to have a little bit more room to play with if you get a perturbation. If that could be used widely, I do think so. Why that is not something we do all the time, because I also think that stability is a little bit the enemy of maneuverability, because it's great that you are inherently stable, but what you also want is be very maneuverable. It's like falling with style. So you want to kind of like, to your next step, you want to kind of fall, but then like graciously catch it and follow on to the next step. Because I think that's something that I really would like to do, is study not the perturbations by itself, but a continuous perturbation, because that is what happens with complex terrains. But that obviously also gets more challenging when you ask questions about stability, because then one step to the next is a recovery stride, but also an anticipation stride. I hope that answered your question.
Yes, thank you.
Okay, thank you very much.